If I look like I'm cold, it's because I am. Yeah, because I'm from Texas. There was something called fog ice on the road this morning, which I've never heard of, but interesting. So, as he said, I'm Liz Haney. I'm a soil scientist. I live in Salado, Texas, just south of Waco, north of Austin. And we have been at Cedar Falls for 2 days doing the big soil health event. So saw many of you there. I'm going to talk a little bit about the Haney test, obviously, because I was asked to. But I gave a talk, I think last week, for some people in Ohio, and they gave me the title of "The Time is Right for Biology." And I thought that was interesting because, when has it not been? Agriculture began around 12,000 years ago, and at that time, we obviously did not have a lot of synthetic fertilizers or any. So what they did was they had diverse crop rotations, integrated livestock. Their manure was their source of fertilizer, so nutrients in the front and then they come out the back. Really until the Haber-Bosch process was developed in 1909, we were very focused on biology. But after industrial agriculture, we've gone more and more with a focus on monoculture, go big, go home. We're going to put as much chemicals on as we possibly can, and it's resulted in some pretty big problems like the giant dead zone in the Gulf, phosphorus contamination of the Great Lakes, and so really focusing on soil biology is the most important thing we can do right now. And it all starts with the carbon cycle. So plants take in carbon dioxide from the air. They pump that carbon dioxide down to the soil. Half of what they take in goes to their biomass and half goes down into the soil. Half of what goes down into the soil is forming the plant roots, and the other carbon is there to feed the microbes. So throughout the ages of soil testing, we haven't really paid attention to this process. None of this happens, obviously, without rain. So what are we going to try and do with our soil testing? We're going to mimic to how the soil responds after rainfall in the field, and not how it responds to really old chemical laboratory methods. And I'm not saying those don't have their place, I just haven't found them yet. That was a joke. So the way we normally test soil is that it's a chemical-based, physical-based system. It's not living, not integrated. We ignore the biology in the soil. We extract the soil with chemicals that it has never seen before, like really, really nasty stuff, ammonium nitrate, ammonium fluoride, hydrochloric acid, things that are really killing everything in the soil that the soil never sees. So when Rick developed the test, he really wanted to mimic, one, what was happening in the field with nature, which is rainfall, right? And plants growing and living. But he also wanted to see how the soil was in balance, measure the condition of the soil, and then determine what we can do to help. So there's a big misperception that the Haney test was actually developed for soil health, but when he first started it, it was so that we could help farmers in reducing their fertilizer applications, okay? So Rick was a farmhand for a long time. He saw people spending lots and lots of money on chemicals, no control, obviously, over the commodity price or the chemical price, what they're getting for that, no control over climate or weather. And that's you're just left subject to the whole industrial ag system there. But what we can do is we can try to minimize the amount of things we have to put in the soil by determining what's actually going on in the soil. So the Haney test, as I said, we use water because it rains water, we extract water extractable nitrogen, which includes water extractable organic nitrogen and inorganic nitrogen, and water extractable organic carbon. And this is how we see the balance in the soil. We use plant root exudates, malic, oxalic, and citric acids, because those are the three most common that come out of crops. And that way we can determine micronutrients and the macronutrients in the soil. We also measure one-day CO2. So we're measuring the soil's breathing, and that determines the microbial activity in the soil. I actually got an email this morning that said, I think that we want the lowest soil respiration possible. And that's not right, because that means your soil is completely dead. I don't know how to argue with that person. So what we're trying to do is measure this dynamic living system. Soil microbes require carbon and sugars to live. That's where they get their energy. They take in oxygen and release CO2 just like we do. And that's coupled with nutrient cycling. Also, increasing the amount of carbon in the soil. Okay, so we had a question yesterday, how do we measure the carbon that's actually in the soil? Right now, we're just looking at a stable pool, usually, but carbon is dynamic, as we saw from that other slide that had the whole carbon cycle in it. So we want to see how this carbon is cycling through the system. So the majority of nutrient cycling is due to the soil being dry and then re-wetting. So if you've noticed, after a rainfall, plants green up. That's not from the rain. That's because the rainfall has activated the microbes, and then nitrogen comes out, goes up into the plant, and makes the plant greener. And we want to see how well the soil is going to be doing this in the laboratory. Previously, this process was not accounted for. We didn't have a way to measure it. Nobody really paid attention. So the Haney test uses a one-day CO2 burst. We dry the soil out, we re-wet it, and then we measure the amount of carbon dioxide coming off of that soil. So the amount of nutrient cycling is related to the soil habitat, where the microbes are living, and how much food they have. If a soil has no air, it has no water space in the pores, if it doesn't have a food source, then the carbon dioxide is going to be very, very low. Your microbial population is going to be very, very low. So everybody asks, what is a good rating for soil respiration? And you don't have to remember any of this, but you can take a picture of it if you'd like. There is no average, because everybody's soil and context is different. Everybody's climate is different. But we do know that with increased respiration, you're going to get increased nutrient cycling. So if we look at between 100 and 200 part per million CO2, there's a high potential for microbial activity, and that means that they're going to be giving off a lot of nitrogen. You get a lot of credit for the nitrogen in your soil. If it goes way too high, then we need to make sure that we're keeping enough carbon in there to keep those microbes fed. So respiration varies a lot throughout the year, and that's another reason why that range is so big on the previous slide. This is weeks of the year. In January, or like right now when it's really, really cold, there's not very much microbial activity. As the soil warms up and it gets more moisture in the spring, the microbes start getting really, really active. They breathe a lot of CO2. Around week 24 there, you can see a dip. That's when the microbes have used and consumed what was left over there, the carbon source that was left in the soil over the winter. And then as your crops start growing and pumping out more carbon to the microbes, they start peaking again and breathing a lot, very active, providing nutrients to your crops, And then as it gets cold and you've harvested the crops, then your CO2 falls off again. So we want to see how these microbes are acting throughout the season. And I'll talk a little bit later about when to test for your purposes. So, as I said earlier, we've been measuring the soil organic matter, the big giant pool. That's the house that we live in. It's like this big room. And then lunch that was served earlier, that is that smaller part, that water extractable organic carbon that the microbes eat. So what's not important is these walls, right? What's important are all the people in here and the food that we're eating and how healthy we are. And that's what we're measuring with the 1-day CO2. To give you an example, excuse me, of differences in the total carbon or the soil organic carbon in the soil and water extractable. We have these 5 different soils. There's one in Maine that's 6.4% organic matter, all the way down in Texas where we live. It's very sad, 1% organic matter. When we measure the water extractable carbon, there's a smaller pool, but it changes. It doesn't exactly match what is happening with the total soil organic matter. So microbial activity, though, follows that water extractable pool, not the soil organic matter pool. Wow, it's getting hot in here now. It'll be like Adam Chappell and sweating. So water extractable organic carbon, you can see that it increases in the soil in Wyoming, and the soil microbial activity increases correspondingly with it. So that's how we know that that's that food source in the soil for the microbes. And that's what we want to measure. With the plant root exudates, we're using a green chemistry. So the extractant mimics the soil solution by using organic acid to solubilize the nutrients in the soil around the root zone. They temporarily change the soil pH and increase nutrient availability. So with these organic acids, we can measure zinc, manganese, copper, sodium, sulfur, magnesium, all of these. Oh gosh, sweating. And we can make fertility recommendations based on this. It's not quite the same as when you get your standard Malic test and get your fertility recs off of that. Those are based on the relative yield that people think they have seen by testing the soil and applying fertilizer. What we're doing is we're realizing the actual values in the soil that the plants are gonna see, and so we can make direct fertilizer recs off of that. This is a sunflower root, right? So Rick and I went out in the field, pulled some plants up, shook the soil off, and measured everything that was going on in the soil right around those roots, because that's where all the action happens. This is a soybean plant, I think. No, sorghum. Sorghum. And that root mass around it, you can see it doesn't have as many fibrous roots. It's not as intense. There aren't as many roots at all, and not as many of those really small ones. So the soil from, right here we have carbon dioxide, so that's the microbial activity. Right around that sunflower, we had high microbial activity. With the smaller root mass in the sorghum, we had less microbial activity. And then the bulk soil is just something without plants growing in it. So without the plants growing in it, the microbial activity gets lower. Correspondingly, the water extractable organic carbon, which is that food source for the microbes, decreases as well. So does the soil health score and the amount of active carbon in the soil. You can hardly see that. So we've been measuring nitrate nitrogen forever. And that's the only thing that we're getting recs off of. But there's a lot of organic nitrogen in the soil, and especially around that root zone. The sunflower seems to really pump the proteins out into the soil and increase the organic nitrogen. So, since we started testing for nitrogen, we've been missing half of the nitrogen in the soil. So there's an additional 34 pounds of nitrogen on average that we haven't been measuring. And we didn't have the instrumentation to measure that before, but now we do. Rick developed the infrared CO2 analyzer to measure the respiration. And then along came the instrumentation where we could actually start seeing the organic nitrogen and the organic carbon. So for the longest time, we've just been looking at that one nitrate nitrogen. Those are all the different compounds of organic nitrogen that are in the soil. And we want farmers to get credit for having that in there. So an example here, we used to call it the soil health tool. We didn't call it the Haney test. Somebody else came up with that. So the soil health tool score here, the green bar, is how much nitrogen we see with the Haney test. The nitrate nitrogen is that smaller orange one. So that difference, the purple bars, that's how much nitrogen we've been missing. And that equates to a lot of dollars. When you get your soil test back, you'll see traditional N, that's that inorganic, that nitrate that we normally measure. Then you'll see the Haney N, and that's the total. So it's got ammonium, nitrate, and the organic nitrogen. The difference in N would be the ammonium and the organic nitrogen. And then you also get the savings in dollars based on how much the current fertilizer prices are. So, on-farm impact. The test is more expensive. It's $55 a sample, but you get a lot more information. You can take one sample per 50 acres. You can go up in that if your soils are very uniform. Need to update these slides a little bit, but the cost is essentially $1 an acre for the test. With an average savings of about 20 pounds an acre, the return on investment is about $2,000. So even though the test is more expensive, just on the nitrogen alone, you could potentially save that much money. So there's the impact of the Haney test is huge. I remember people coming to visit Rick at the lab. They'd be standing there and they'd be astonished to think about how much nitrogen fertilizer that we would be saving just by using the Haney test. It makes the fertilizer companies really, really happy. But the Haber-Bosch process to produce nitrogen is very energy consumptive. It accounts for, I think, 1 to 3%, depending on who's quoting it, of all of the energy consumption in the world. So if we can reduce the amount of nitrogen that we're putting in the soil, the synthetic part, that would be equivalent to greenhouse gas emissions from about 50,000 cars per year, if we just did, about 4 million acres. So, as your soil health increases and the microbes are more active and the biology is happy, it's going to be cycling those nutrients. We can measure that with the soil health calculation. And as that soil health calculation comes up in the Haney test, the dollars of nitrogen saved per acre also increases. And I don't remember how many samples are in this, but that's a lot of samples. So we get a lot of questions on how to use the Haney test, and it really depends on what your goal is. Are you looking to reduce your input costs? Are you trying to figure out how much carbon you're sequestering in the soil? Are you trying to determine how healthy your soil is? We can look at the soil test and see if you have low microbial activity, we can pretty much assume that you're not getting water infiltration. You don't have water holding capacity in the soil and not a lot of retention there. We can demonstrate the impact of management practices on soil biology and nutrient cycling. and assess your progress over time. So, when to test? What is your goal? Do you want fertility recommendations? Do it right before you're going to get the fertilizer. Do you want to monitor soil health throughout the years? Take it around the same time, the same temperature, same moisture, year after year after year. If you take a sample in the spring and then take another one in the fall, you're going to get two different numbers. So in the spring, you haven't had your crops going, maybe you just put your cover crops into the ground, you're probably going to get good respiration, like I showed you on that slide earlier. Your soil health scores will be higher, and then as your crop grows, after harvest, you're going to get lower values for nitrogen, phosphorus, just because of the uptake of the plants. So if you want to measure your soil health, establish a baseline, start one time a year, come back the next year, do it again, and we can see how the health of your soil is improving. So another way to use a test is we do product trials. And this is one example where we had a plot with corn. This is Russell, he's a short guy, no, he's really, really tall. The plot on the left, yeah, your left. The plot on the left was without an application of algae, and the plot on the right was after an application of algae. So we saw increased plant growth, but what did it do to the soil? So we were able to measure and see that microbial respiration was increasing, bacterial populations were increasing, fungal populations were increasing. So it's a very good indicator of what a product is doing to the biology. So I mentioned you can see how your management practices are impacting the biology of the soil, and we can test that and see. So year after year, this is again Russell's soil, the percent change from when he just started doing no-till without covers to moving into using cover crops, he had a 720% increase in soil respiration, a 590% increase in the water extractable organic carbon, which is that food source for the microbes, and his soil health score increased by 74%. His organic matter, his total organic matter, increased by 5.6%. So we're looking at the soil from baseline to after implementing practices, and we keep seeing increases in these indicators year after year after year. Like I showed before, this is the part of the soil test information that you get, but this is the same soil, and as his soil health is increased, he's been able to increase the production of organic nitrogen, and here he has 187 pounds of nitrogen available. If you have no biology, that number is going to shrink. So the way we're using this now with management practices, Rick and Lance developed some equations that show the soil health from 0 to 6 and the soil health from the 6 to 12-inch sample, and we can see improvements based on management. So we have a verification process. It's scientifically validated through this soil testing. It's climate, soil, and context specific to your operation because we're comparing your management to your management. The baseline, the 6 to 12-inch, and the 0 to 6 is where your management is impacting the soil. And with their calculations, we have developed where we can see regeneratively grown foods are coming from. So the soils on the top, we know they've been using regenerative practices. They've been increasing microbial activity, they've been using cover crops, implementing livestock on their land, and gosh, it's been a long day. the soils on the bottom come from more conventional farms. So when they run their check, we can see which ones are being regenerative through their management practices, and those would be above that 6.8 line. This is an example of our verification. I hope y'all can see that. The top line is a 0 to 6 inch, and the bottom line is a 6 to 12 inch. So we see that this person in the 0 to 6 has more nitrogen, more organic nitrogen than in the 6 to 12. They have better respiration right here in the 0 to 6 than the 6 to 12. They have more organic carbon, which is more food for the microbes, in the 0 to 6 and the 6 to 12. And they have a good C to N ratio. Their soil health score is higher in the 0 to 6 than the 6 to 12. So we can tell that these people are regenerating their soil by the management practices. So ways that we're using this, this is Dave Brandt out in Ohio. We tested his soil. He's growing grains, milling them, and selling them around Columbus and that area. And they want that because it's regeneratively grown, it's regenerative verified, and it's got increased nutrient density. So he's tested his protein contents, and they're much higher than in a neighbor's crop where they've been conventionally tilled. We call him the godfather of soil health. He's been no-tilling since, I think, 1970, way before I was born. We're using his products in making beer, and like I said, the milling stuff. And what we're really trying to do with our regenerative verification is make the consumers aware of regenerative practices that improve the soil, improve water infiltration, improve water holding capacity, reduce nitrate leaching, reduce phosphorus running off of the soil, improving water quality. And the way that most people, to get to their heart is through beer or steak. This is another one of our growers out in Darty, out in Tennessee. He and his sons grow, I think, 5 or so acres of a sweet corn every year. And we tested their soil, it's regenerative verified. And he has a label on there. So when he and his sons go to their farmer's market, they can sit there and tell them, look, we tested our soil. We can tell we're improving that 0 to 6 inch zone through our practices. And so by what we're doing, this corn that you're buying from us is improving the soil, improving water quality, and better for the environment. With our testing, like I said, on Dave's wheat and stuff, he has higher protein content. This corn right here was tested. It has 12.8% protein. So it was grown using regenerative practices. We tested the soil. It's regenerative verified. And this grower is getting a premium at selling his corn to a feed mill. And then we're also making bourbon out of it. So the higher protein content and the higher starch content in this regeneratively grown corn means that we can actually make more liquor out of it with less grain. And it has like a really earthy flavor. People tell me I don't touch the stuff. No, I'm just kidding. But it's another way for the consumer to see that what you all are doing is making an impact on the environment. And I think it's really important for them to know that the practices that you're doing are not killing them. I've heard farmers say, everybody thinks that we're trying to kill them. No, we know you're not. So we want to tell that story. So our next step in that is that we are working with Transparency Wise, and we are going to make it where a consumer can go in and see our certificate on the meat product that they're buying, the flour they're buying, the vegetables they're buying, and know with a completely transparent method that they've had their soils tested, they're improving the soils using the Haney test. We can see that, and the consumers will become more and more aware so that the farmers can get a premium on the products that they're growing using these responsible practices. I did that really quick. Does anybody have any questions?
On the slide that you had the algae applied, the guy standing there, at what stage was that applied? In furrow, or was it?
He did in furrow and 2x2, and then foliar application.
What is your criteria for how often you use the Haney test? What pattern or how many acres you test, etc.?
So it's going to depend on your goals. If you do want to know, let's say you're in a corn soy rotation, you're going to want to do it before you get the fertilizer for your corn so you can make a good fertility recommendation. Before you plant your next crop, you're going to want to test it again. If you're doing side dress applications, you can take them before side dress as well. And as far as like acreage size, if you have a pretty consistent soil, you could do 1 sample per 50 acres up to 1 to 100 acres. If you're doing precision application, doing the Haney test on 2.5 acre grids is going to be a little cost prohibitive. Yeah. So what we do is maybe do a composite sample on the whole field and measure the organic nitrogen coming from the Haney test, and then do your normal grid testing. And once you get that, you can subtract off from your fertilizer recommendations the amount of additional nitrogen that you're seeing from that composite sample. Anybody else?
Do you recommend a lab?
Yes, Regenerative Ag Lab. Thank you. They're the only ones that have the original founder actually working for them. All right, thank y'all.